12th Bio-botany Chapter 4 Principles and Process of Biotechnology. Under the heading Molecular Techniques, in the previous video we discussed about gel electrophoresis. In this video we shall discuss about nucleic acid hybridization and blotting techniques. What is blotting? What is the use of blotting? Blotting techniques are widely used for the specific identification of desired DNA or RNA fragments from large number of molecules. When we have more number of DNA fragments, we can identify a particular DNA fragment by means of blotting. Actually, what is the meaning of blotting? In blotting, macromolecules like DNA is transferred from gel to the solid support nitrocellulose or nylon membranes and immobilized. In blotting technique, DNA is transferred from gel. In the previous video we discussed about gel electrophoresis. In gel electrophoresis, DNA molecules are separated based on the length. This DNA on the gel are transferred to solid nylon cellulose, nitrocellulose or nylon membranes and fixed. This is blotting technique, transferring of DNA pieces from agarose gel to a solid support and fixed. The blotted nucleic acids are then used in the hybridization experiments for the identification of desired DNA fragments from large number of molecules. The desired DNA fragments are identified in hybridization experiment only. What is hybridization? Two single-stranded DNA molecules are combined together to form a double-stranded DNA based on base pairing. When the base pairing of two DNA fragments are same, when the two DNA fragments have complementary bases, the two fragments pair together and form hybrid DNA. We shall discuss about the hybridization elaborately later. These are two DNA fragments. Both are single stranded. They have complementary bases, so they combine together and form a single DNA. This is hybridization. Hybridization is the process of combining two complementary single-stranded DNA or RNA molecules and allowing them to form a single double-stranded molecule through base pairing. The hybridization is based on base pairing. There are three types of blotting: southern blotting, northern blotting, western blotting. Southern blotting is used for blotting DNA. From the agarose gel, DNA is transferred to nitrocellulose membrane. In northern blotting, RNA is transferred from agarose gel to nitrocellulose membrane. In western blotting, proteins are transferred to nitrocellulose membrane. Southern blotting, northern blotting, western blotting. Southern blotting is for DNA blotting, northern blotting RNA, western blotting proteins. In this part we are going to discuss about southern blotting in detail. In southern blotting technique, it was introduced by Southern in 1975. What is done? DNA on the agarose gel is transferred into nylon membrane for hybridization. A long DNA, total DNA, a part of the DNA is the needed gene or gene of interest. So the DNA is cut into fragments by restriction enzymes. One of the fragments have the gene of interest or the gene that we need. The DNA fragments are transferred into agarose gel during gel electrophoresis. The fragments are separated by gel electrophoresis. We discussed gel electrophoresis in the previous video. This is gel electrophoresis technique apparatus. DNA is negatively charged molecule, so it moves from negative electrode to positive electrode. The DNA fragments are introduced into the wells. When it is switched on, the DNA molecule is negatively charged molecule. Under an electric field, DNA molecule migrates through the gel, and as the DNA molecules migrate through the gel, banding patterns are formed. So after gel electrophoresis, the DNA fragments are separated, and the DNA separated fragments appear as separate bands on the gel, agarose gel. The gel is soaked in an alkali solution to denature double-stranded DNA. So all the bands have double-stranded DNA. For hybridization experiment, we need single-stranded DNA. The separation of two strands from double strand is called as denaturing. Denaturing is done by alkali solution. The alkali solution breaks the hydrogen bond between two DNA strands. So DNA gets denatured and two single-stranded DNA are formed. The denatured DNA fragments are now found on the agarose gel. This agarose gel is then placed on a platform in a dish containing buffer. This is a dish. Buffer is added. Now buffer is added. This is a platform, a glass support. Over the glass support, a filter paper or blotting paper is fixed. This is the edge of the blotting paper. Let us see the surface view of the blotting paper and how the blotting paper or filtered paper is placed on the glass. This is a glass plate. The blotting paper is placed on the surface of the glass, then both the edges are folded. This edge is folded, another edge is folded. Now the edges of the blotting paper touches the buffer kept inside the tray. Due to capillary action, the buffer is drawn upwards. The buffer reaches the surface of the filter paper. Here the filter paper acts as a wick. In chimney lamps, the wick thread draws the oil to the top. Here also due to capillary action. The buffer is drawn upwards. So this is the edge of the filter paper. Above the filter paper, what is kept? The gel containing single-stranded DNA fragments. Denatured DNA fragments are on the gel. That gel is kept here. This is gel. The buffer is drawn upwards by the filter paper, and the buffer passes through the gel. The DNA dissolves in the gel, so the DNA fragments also move upwards as the buffer moves. Then a nylon membrane is placed on the gel. The DNA moves as the buffer moves, and the DNA is fixed on the nylon membrane. The nylon membrane attracts the DNA. It has affinity towards DNA. So the DNA does not passes beyond the nylon membrane. The moving buffer alone passes through the nylon membrane. That is blotted by blotting paper or filter paper. Excess buffer is drained by paper towel or tissue paper. Then a weight is applied. When we keep a weight, due to pressure, the buffer moves upwards. Let us see the outline of the setup again. A glass support. This is blotting paper. It draws the buffer moves upwards. Gel containing denatured DNA fragments. As the buffer move upwards, the DNA also move upwards, and the DNA is fixed on the nylon membrane. DNA is fixed. The moving buffer alone moves beyond the nylon membrane, and it is blotted by blotting paper. Excess buffer is drained. This is weight. Now the DNA is blotted from agarose gel to the nylon membrane. The next aspect is hybridization. The membrane containing denatured DNA fragments are kept inside the hybridization bottle, and probes are added, radioactive probes are added with the denatured DNA fragments. DNA fixed on the nylon membrane is placed in a hybridization bottle. The bottle is filled with solution that contains radioactively labeled probe. Probes are short DNA fragments. We know the DNA sequence of the probe. The probe is radioactively labeled. Then the bottle is gently rotated. When the probe DNA sequence is complementary to any one of the single-stranded DNA, the probe binds and form a double-stranded hybrid DNA. When we rotate the bottle, this happens. What are probes? Probes are single-stranded molecules or radioactively labeled or fluorescently labeled DNA or RNA. Here red color indicates the probes. The probe binds to the gene of interest that we want to detect. We have to find out the gene of interest, or we have to separate the gene of interest. For that, the probes are used. The probe binds with a complementary DNA fragment. This is a probe. Its sequence is ATCCGA. The probe is mixed inside the hybridization bottle with other DNA fragments. These are DNA fragments. The probe DNAs are mixed, and the probe combines with the complementary DNA. This is our gene of interest. Base pairing indicates the gene of interest. The probe sequence is known to as, and the gene of interest, this is gene of interest. It is complementary to the probe, so probe binds and form a hybrid DNA. The probe binds to the complementary DNA fragments on the membrane and form hybrid DNA. So hybrid DNA, in 3 areas hybrid DNAs are formed. They are double-stranded DNA. Autoradiography detects the fragments with probe attached. In which area the probe is attached and form a hybrid DNA. That can be detected by autoradiography. Autoradiography shows the hybrid DNA formation. The probes are attached with the complementary DNA fragments and form a hybrid DNA. The DNA hybridized with the nucleic acid can be detected by autoradiography. What is done? The nitrocellulose sheet or nylon sheet is placed over X-ray film. X-ray film darkens where the fragments are complementary to the radioactive probes. In this way, we can identify the needed DNA pieces from the group of DNA fragments by Southern blotting technique. And in gel electrophoresis, already we discussed in the previous part, marker DNA fragments are introduced into one of the wells. The DNA, sample DNA is compared with marker DNA to find out the length. So this is Southern blotting technique. If you understand this one, if you read the book, you can understand other 2 blotting techniques.